It was not so long ago that DNA replication was a complete mystery. If it's still a bit of a mystery to you, don't worry. When I was a freshman, I didn't completely understand DNA replication either. Um, so, although it's common knowledge to biologists today, there were three differing hypotheses just in the 1950s, not so long ago. The conservative, the semi conservative, and the dispersive models, which we'll talk about later in this speech. The scientists who proposed these hypotheses, however, did not have evidence to back them up, and so these were merely speculations. Although these models, starting with the conservative DNA replication model, this one shows that the parent helix would somehow remain entirely intact and that the daughter strand would be made of completely new synthesized nucleotide sequences. And there would be a completely new DNA strand. In the dispersive model, as you can see, the strand would be copied in short segments and there would be no unwinding. There'd just be segmenting and adding nucleotide sequences. In the most widely accepted model at the time was the semi-conservative DNA replication model, which shows that that each new DNA strand consists of one of the original and one newly synthesized DNA strand. And so here they all are. If you think you need to, you can write the differences down, or you can write these down on your paper, since I'll be referring to them quite often. Two scientists, Nesselson and Stahl, began research in Rochester, New York. Began researching the three hypotheses of DNA replication in Rochester, New York. Their research goal was to figure out which of these hypotheses were correct. They accepted that one of these were correct, and so no outside hypotheses were correct. And so they designed an experiment to test this theory, or to test this hypothesis. The results may not shock you. Although it took several years to design an experiment that would provide this answer they needed, it turned out to be the most beautiful experiment in biology. In 1952, scientists discovered that DNA is the genetic material in cells. Shortly after this, two scientists named Watson and Crick discovered that replication occurs in DNA. People wanted to know, like, how does replication occur? Since DNA is the building blocks to life, they wanted to know how it worked. I mean, why wouldn't you? And so this is why it's important to us. After these years of research, the experiment started by growing two batches of E. coli, one in a nitrogen-containing isotope of nitrogen-14 and the other in nitrogen-15 isotope medium. The DNA in the heavy isotope was then added to the light medium. These resulting strands were then broken up by heat. Each of these resulting strands were then put in a density centrifuge. Why? The E. coli were added to the different nitrogen isotopes because the nitrogen is a major constituent of DNA, and so it'll make up a lot of the DNA's mass, which means that the nitrogen-14 will have a lighter. The DNA with nitrogen-14 medium will be lighter than the DNA in nitrogen-15, which will be a heavier DNA, which is how they distinguish between the two. The density centrifuge was used in order to read the density of each of these nucleotide sequences. And so the heavier DNA was then added to the lighter medium in order to rule out one of these hypotheses. The resulting strand was then heated in order to decide which of these last two hypotheses were correct. And so, the resulting strand added to the results show that the strand of DNA added to the lighter medium had a density in between that of pure heavy and light DNA, which rules out the possibility of the conservative model. As you can see here, the conservative would show two distinct densities. One of the completely newly synthesized strand, which would be made of the light isotope medium, and one of the parent strand. And so they would have two density lines. And in this one, there was just one directly in the middle, which rules out this possibility. Afterwards, this resulting strand was heated and detached from the double helix, and the strands were then measured in the density centrifuge. The strands were shown at two distinct densities, each identical to that of the heavy and to the light isotope containing DNA. And so this ruled out the dispersive model. As you can see, the dispersive model contains both newly synthesized and parent strand within one strand. And so as they're split, they would still have one density line directly in the middle of the light and the heavy isotope. In the semi-conservative model, however, as the two were split, there would be a parent and there would be a newly synthesized strand, each at two distinct density points, which was seen in the results. This means that the semi-conservative model is the accepted model of DNA replication. Since there are only 3 accepted hypotheses for the process of DNA replication, ruling them out one by one seemed to be the most proficient way of discovering the truth. These results, however, could have been stronger if Stahl and Meselson accepted more hypotheses into their experiment, though it's not argued against widely today. There have been more studies done after this experiment, and so unlike how replication can be different within certain specific cells, and so DNA replication experiments are still going on today. So Stahl and Meselson accepted that one of these hypotheses that I've mentioned in this speech were correct, and their experiment was directed toward ruling these models out one by one. And so the results showed that the heavy DNA added to the medium or to the light medium showed one distinct strand, and that these strands being unwound showed two distinct densities. This tells us that the dispersive and the conservative models can be ruled out, and so the semi-conservative model is the accepted model out of these 3, is the accepted model of DNA replication. Their main research goal was to discover which of these 3 were the accepted truth of DNA replication. And since they ruled out the conservative and the dispersive models, their goal was reached their goal was satisfied. Up till today, few arguments have been made against this, and no evidence has proved otherwise that this experiment was false. Thank you.